So let's move on to vagina. Here we find the stratified squamous non-keratinized epithelium. There is a new term, stratified. We discussed the term stratified, which means there are many layers of epithelia. Here the new term is the non-keratinized. Non-keratinized indicates that there is no presence of keratin inside the structure. What is keratin? What is the unique feature of keratin? Keratin, of course, is a filament that we find in the skin, of course, and the purpose of keratin is to prevent water loss. In fact, this is also one of the elements that is used in the Darwin's evolutionary theory, that keratin was actually the item, the protein, the content that helped us escape the sea and progress this and move out to the land. The major problem, of course, with this movement, this transition, was the water loss, because the bodies, the ancient organisms were actually used into massive amount of water inside them, with massive quantities of water surrounding them. There was no need, there was no presence of the X-rays dehydrating action. So because of keratin, because of this dry substance, because of the presence of keratin, it allowed from the organism to transition out from the sea and go to the land. So the point, generally speaking, of keratin is to simply prevent water and liquid loss. So generally speaking, whenever you see the term non-keratinized, you should immediately think that this area is wet, from tears, from whatever it is, saliva, vaginal secretions, whatever it is. So the point is that because we have the non-keratinized, it means that this area is wet and there is no need, there is no fear of dehydration, because the substance, because of this content, this cell is surrounded in liquid areas. So even if there is loss, there's going to be easy reabsorption and there's going to be a proper maintenance and balance of liquidity within the cell. So let's see, actually, we just discussed about this is a very, very important thing that I should actually mention. When you name a cell, when you give a specific term to the epithelia, you should have a specific sequence. It's not just random terms. You should specify these terms in a very specific way. First, you mention the number of layers. So we said, for example, simple columnar epithelium. Again, simple is for the number. Then we talk about their shape. They can be squamous, they can be cuboidal, they can be columnar. So, for example, before we said simple columnar epithelium. And the third one is the keratinized and non-keratinized. For example, in this case, we find the stratified, squamous, non-keratinized epithelium, always in this sequence. So let's talk about this stratified, squamous, non-keratinized epithelium. So of course, this is a section of the zoomed out of the vagina, and if we zoom in the surface and the covering epithelium, of course, we're going to find many different layers. So we'll discuss these layers much more detailedly in the skin in a bit. But the point is, we can easily distinguish many different layers of cells, and how to easily distinguish keratinized from non-keratinized epithelium. The trick is in the presence of nuclei on the top layer. So the top layer right here contains clear alive nuclei. So of course, this is the easiest thing to say. This is the non-keratinized epithelium. Let's see in comparison how the keratinized epithelium looks like. So this is the zoomed out appearance of the vagina. We should zoom in towards the epithelium, towards the top. And again, we can easily understand the stratification because there are many layers of cells, true layers of cells. So for example, these cells, the intermediate cells, are not connected to the basal membrane in no way, and we can actually clearly distinguish the different layers. And most importantly, we can distinguish the difference in the non-keratinized and keratinized from the presence of nuclei on the top layers, such as these right here. The skin is the perfect and ideal example of the simple squamous keratinized epithelium. Why do we care so much? Well, because here we can find the typical appearance of this keratinized epithelium. There are specific layers. We should discuss about these layers. First off, we can find the Stratum Basale, the basal layer. As we said before, whenever you hear the term basal, your mind should directly go into the germination, practically to the mitotic rate, practically to functions that are of mitosis, and practically structures that are responsible for the regeneration of the structures. So here we find the first layer, the Stratum Basale, these cuboidal looking cells, and these cells are found are actually resting on the basal membrane, and these cells are again responsible for the regeneration of the skin of the keratinocytes. So this is the first layer. The second layer we have is the stratum spinosum. What is the stratum spinosum? The term stratum spinosum is the spinal layer actually is very, very helpful. Why? Because here this is the first example of the production of keratin, and here we find the first cohesion, first adhesion of these keratin molecules from the single cell to the other cells. So this is where the cells of the stratum spinosum start to produce this keratin, the substance of keratin, and because it's practically filaments, they start to connect. They're connecting points of keratin from one cell to the other keratin of the other cells. So these points, imagine having a cell with many different points of keratin. So if you start pulling them or contracting them, because this is the result, if you connect two structures, if you connect two different points from one cell to the other cell, these points of connection and these points of binding will actually start pulling and pushing the cell in many different ways. So because you have many different points of this pulling motion, you're going to have this appearance of this spine. That's why it's going to have this polyhedral shape, and this is because of this connecting point of the keratin between the different cells. So this is why we actually call it the stratum spinosum, because this have the appearance of the spine, which is caused with the connection of keratin from many different cells all together. Next up, we're going to find the stratum granulosum, and here we're going to find cells that actually are full of granules. This is why it's called granulosum. In fact, we're going to find two different granules here. One type of granule actually is going to produce molecules and proteins that assist with the keratinization. The second one is going to be full of lipidic, rich in lipids granules, which will be exocytosed. Why do we need to have lipids in this structure? Well, because the point of skin, the point of the function of the skin is to prevent dehydration. So what molecule is ideal for pushing away water? Lipids. Water is lipophobic, so there is no tendency of water to adhere or to practically go towards the lipids. So by adding this layer full of lipids, you the cell induces this water, the inhibition of this water movement. There is no tendency of water to go outwards simply because there is a layer full of lipids pushing it back in this way and not allowing it to progress furthermore. Actually, the lipids play a dual role in this case, both for water to go in and water to go out. So just imagine this is this layer of lipid insulation that doesn't let water pass through easily. Of course, we know clinically speaking that water does pass through in and out, but it prevents the major components and the major losses of water or major gainage of water. So this is the function of the stratum granulosum. Next up, we're going to find the stratum lucidum. The stratum lucidum is not found in all of the skin, only in very few areas. Actually in the hands, the palms, and the soles. These two components, just by if someone tells you that the thick skin is found only in two layers, only in two locations of the body, the palms and the soles, and this layer is found only in these layers, you immediately have to assume that there is some sort of correlation between these two functions. And indeed they do. Because the stratum lucidum is found in the palms and the soles, the scientists actually have concluded that this layer is nothing more than a result of mechanical compression and friction between these surfaces. Of course, because the palms and the soles receive the highest amount of mechanical irritation and friction. So, and this is where we can find the stratum as well. And lastly, we find the stratum corneum. This is the most apical portion, the most top part of the skin, and this is where we find literally dead keratinocytes. These cells that are filled with keratin, they contain no nucleus, they contain nothing. They're just dead cells with only an excessive amount of keratin in their content, in the cytoplasm, and these keratinocytes are actually connected through this intercellular connections between the one cell and the other, connecting all the keratins from all the different cells into practically one uniform layer that is called the stratum corneum. Let's visualize in the microscope. So this is again a very nice section of the skin. Just to mention, might find there are two different types of skin, the thick and the thin. The thin is the one that does not contain the stratum lucidum, and the thick is the one that contains it indeed. So here we can start seeing these different layers. Of course, the first layer is always the stratum basale, Then we're going to have the Stratum Spinosum, which has this spinal appearance of cells. Then is the Stratum Granulosum right here, this full granular layer. And lastly, the Stratum Corneum on top. This is another section, practically it shows exactly the same thing. Basale, Spinosum, Granulosum, Lucidum is not found here, and the Corneum as it is. So this is the thin skin. You can actually see that again we have this keratin on top and a lot of basal cells of course in the present again, and the point of the thin skin is that we find way fewer, of course, cells, way fewer, there get way thinner keratinocyte, and there's continuous and absolute absence of stratum lucidum. And look at the difference. Look at the thickness of the thick skin, and look at how thick is the thin skin. And this is actually simply to denote there's a difference in their function. The location of the same structure, the location of the same exact tissue has different morphology because of the different functions. Always speaking, the structure denotes and defines function, and vice versa, and function can define or somehow alter the structure. Just because we're in the human body, and just when the body is generally speaking in a live tissue, the different conditions, the different function of this tissue is going to be reflected in their structure. And if you just see it by a structure, just analyze the structure, you can actually start having very accurate hypotheses and very accurate assumptions over its function. So let's talk a bit about how it looks like in the microscope. Again, first off, we can find the Stratum Basale right here, very close to the basal part and connected to the basal membrane always. Exactly after, we're going to find this very, very big layer of the Stratum Spinosum. And again, here we can find these spinal looking cells, which again, this is because of the increased amount of keratins being pulled in the many different locations of the cells, hence causing it to have this spinous appearance. Then we're going to find the Stratum Granulosum. This is the Stratum Granulosum right here. And in fact, we can actually see this much clearer in the second section. So, for example, this is a much clearer section of the Stratum Granulosum. We can see the granular appearance of the cells. We can see that these granules are the ones that we're talking about. There's two different types of granules. And lastly, we have this right here. This is the Stratum Lucidum. This is only found in some locations of the thick skin. And this is again as a consequence of the mechanical compression of the cells. And lastly, we find this very, very large Stratum Corneum filled with keratin fibers. And of course, all of these, there is no presence of nucleus. This is the typical characteristic of the keratinized epithelium on the top in comparison to the non-keratinized. And these cells again are filled with keratins connected to each other, preventing water from coming inside or coming outside. Thank you so very much. So lastly, we should talk just a bit about the glandular epithelium. We're going to go through the glandular epithelium much more in histology too, but this is just a short, let's say, introduction about the glandular epithelium and how it works. So the glandular epithelium actually comes from the covering epithelium. This is the production, how it's being genetic, the genesis of the glandular came from the budding of the epithelial cells, the covering epithelial cells. So there are two different ways that this can happen. In both ways of the endocrine and exocrine glands, exocrine means is the structure that is the gland that is connected and open to the surface that it's connected with. Typically, for example, let's say in the skin, we have sweat glands. These sweat glands are exocrine glands because the sweat is produced by the gland that's on the bottom, and on the top there's an actual communication from the point of origin to the point of deposition. And in the case of the endocrine glands, the endocrine means the production of molecules or signaling molecules towards in the blood, and from the bloodstream towards to the point of targeting. But both of these actually glands come from the covering epithelium. And of course, for the first action for this formation is this budding, the inward budding of the glandular epithelium. So we have this inward budding. If this duct is maintained and actually stays there, this is the formation of the exocrine gland. If the duct cells disappear or disintegrate or die for whatever reason, then this gland is going to actually end up as an endocrine gland. Typically speaking, we should mention a bit about like the typical function, the typical structure of any glandular structure. So first we have the acinus. The acinus actually comes from the Latin term acinus, which actually means grape-like structure because it actually looks like a grape, and the duct portion. Always speaking, typically speaking, the acinus is going to produce whatever surface, whatever liquid or whatever product it is, and typically, always speaking, the duct will tend to modify this liquid. And this is a very, very important feature. Why? Because typically speaking, the first product, the first cellular product is going to contain excessive amount of water or excessive amount of ions. So in an attempt of our body to save ions, to save water, and as a consequence to save energy, this is the function of the duct, to practically reabsorb water, to reabsorb ions, or in some cases to enrich ions and to enrich substances in the product. There are many different morphologies that you should see. This is just a picture again from Jonquera. This is just for you to see that there can be many different morphologies of the, many different shapes of the glands and many different organization techniques of the glands. There's no point in discussing them. Just take a look at this table and just have a wider picture of the glands and how they work. We're going to find three different types of secretions, generally speaking, in the glandular epithelium. There is the merocrine, holocrine, and apocrine. Merocrine means, mero comes from the word mer. You already heard this term, monomer, polymer. Mono means one, mer is part. So merocrine is only part of the secretion. Specifically, in the merocrine glands, we find this, typically, of course, we find the granule formation and the granular exocytosis. So the cell produces this product that you want to produce, and for example, many different enzymes or whatever it is, and these contents are being exocytosed from the granules towards the duct, and the duct, of course, gives out the content. The second one is the holocrine. Holo comes from the word whole, which means comes from the word holo, which means whole. So this point is that the whole cell eventually becomes the point of exactly the same content of the product. So the cell becomes itself the product. Typical example is the sebaceous glands in the skin, which produce sebum, which is a rich liquid, rich in lipid-rich content, and practically the whole part of the gland, the whole cell, the cellular epithelium of the cellular gland is going to become the product. So this is the very first example of the holocrine gland. Typically speaking, the more liquids, the more lipid one content contains, the more parts, the bigger part of the cell membrane is being disintegrated with it. I'll come back to this in just a second. In the last one, we can find the apocrine gland. This means that the apical portion of the cell buds off and buds out towards the and becomes a secretion. In this, for one typical example, is the mammary gland here. Mammary gland also contains lipids, of course. Primarily proteins, but also lipids. Generally speaking, the content will somehow indicate a change in the different type of secretion. In what way? Well, in the apocrine gland, we typically have a mixture of the content is typically speaking, not always, typically speaking, a mixture of lipids and proteins. Typically in the holocrine gland, the whole product is lipid and takes away all of the cell membrane. And why do we care about this? How is this connection relevant? Well, because the amount of cell loss is proportional to the lipid within the content. So if the content is full of lipids, there's a high chance you're going to lose the whole cell or part of the cell. If there is a lower amount of lipids, it's more typical and more common that a part of the cell membrane, which again is phospholipids, again contains lipid product, will typically bud off with it. That's why in the apocrine gland we find this unique epithelium called we usually tend to call them low columnar to cuboidal. Why is that? Because typically the cell, if this wasn't a productive cell, it would be a columnar. But because it produces a mixture of lipids and proteins, as a consequence, it will have this budding off of the top part and will have this low columnar to cuboidal appearance. And this short, practically cell is a consequence of this lipid component budding off along with parts of the cell membrane with it. Thank you.